This presentation will review the concepts of fluorescence as used in cell culture. Fluorescence describes a property exhibited by some molecules termed fluorophores that absorb one wavelength of light, a shorter wavelength, and they emit another wavelength of light with a longer wavelength, as shown in this figure where blue light is absorbed and green light is emitted. This figure on the right is illustrating the electromagnetic spectrum with the visible spectrum enlarged at the bottom, with relative wavelength shown here. Note that the wavelengths use the unit nanometer, which is 10 to the -9 meters. Fluorophores are very useful for scientific applications and come in a wide variety of options. Fluorophores are often visualized with special microscopes set up for this purpose. These microscopes will have filters that allow the excitation wavelength to hit the sample and another filter to allow the emission wavelength to be detected. One of the biggest problems with this type of microscopy is autofluorescence. This is where the sample has molecules within it that will fluoresce even in the absence of the added fluorophore. Obviously, this is a problem for samples in an experiment and will require alterations of your protocol. Another problem is photobleaching. This is a phenomenon where prolonged excitation of the fluorophore with light causes it to stop fluorescing altogether. This results in fading of your sample over time. Thus, when working with fluorophores, you must protect them from light until you're ready to image them, or you risk losing sensitivity, or worse, risk losing all of the signal from your sample. It's possible to stain samples with multiple fluorophores that stain different structures, and then have the computer stack the images taken with the different filters to visualize the multicolored staining shown as shown in this image over here. The green looks to be staining the actin and the cytoskeleton. The blue is staining the nucleus, and the red is staining an organelle. With specific protocols, it would be possible to stain living cells over time and observe changes occurring within the cells. This slide lists a few of the common fluorophores, but there are very many available. For example, DAPI emits blue light when excited and is used to stain the nucleus because it intercalates into between the DNA strands. The host stain also binds DNA and emits a blue-green color. Calcin and fluorescein, also known as FITSI, emit a green color, and propydium iodine emits red. This image on the right shows the color emitted by quite a few of the different fluorophores available commercially, but there are many more as well. Antibodies, proteins made specifically by our B lymphocytes to bind foreign molecules, are very useful for visualizing cells with particular molecules because they can be coupled with fluorophores. They are used diagnostically to determine if a patient has antibodies to a specific antigen. Antibodies that are coupled to fluorophores are often monoclonal. I'll describe that process in a moment. Note on this figure that you have green antibodies bound to specific molecules termed antigens on a cell such as a bacterium. Different molecules are depicted by different shapes, and they're bound by antibodies specific for that particular molecule. Monoclonal antibodies are available commercially, and they're produced by hybridoma cell lines. Making a specific hybridoma is quite involved, and this figure tries to simplify it visually for comprehension. First, you immunize the mouse with the protein of interest, the protein you want antibodies to, and next, the mouse immune response kicks in, and B cells producing the right antibodies multiply in the mouse and start releasing antibodies to that protein. The B cells are isolated from the mouse. This usually involves removing the spleen from the immunized mouse because there are large numbers of B cells that live there. The B cells are then fused with cancer cells to make them immortal. Not all the cells survive this process, but some do. After this, you must dilute out the surviving cells and screen for the ones that are making the correct antibody. Once you identify the correct cells, you can expand them into a large culture vessels and purify the antibodies they're making. Now you have a monoclonal. Mono meaning one and clonal referring to the type or clone of B cells that you're working with. The ones making your antibody. This slide is demonstrating how antibodies are used to detect something in the cell. This figure here on the left shows a monoclonal that's been linked, called conjugated, to a green fluorophore. The antibodies bind the protein of interest, and now the cell will glow green. This second image, the middle image, shows that there's a specific antibody bound to the protein on the cell. The antibody is not linked to a fluorophore, so you would not be able to visualize it. This may be a patient antibody. It's not monoclonal. The second antibody is binding the first. This secondary antibody is commercially available. They will bind human antibodies and are linked to a fluorophore. Secondary antibodies are made by immunizing an animal, such as a goat, with purified human antibodies, and the goat will respond with its own antibodies to the foreign protein. The goat anti-human antibodies are isolated from the goat serum and linked to a fluorophore and marketed. Secondary antibodies are useful for detecting patient antibodies and have other uses as well. Once a secondary antibody binds, the cell fluoresces green, so now you know the patient had antibodies that bound. This last image on the right illustrates this idea again. If you only need one conjugated monoclonal to detect in your experiment, you have a direct test. If you need two antibodies with the secondary antibody detecting the binding of the first antibody, then you have an indirect testing situation. And finally, this last image of green glowing bacteria is shown detected with a fluorescently tagged antibody. I hope this presentation on fluorophores has been informative.